Welcome to the Maine Science Podcast. I'm Kate Dickerson. Today's episode is a conversation I had with Jacqui White, the director of the Center for Biometric Analysis at the Jackson Laboratory. I met Jacqui when she moderated the 2024 Science on Tap session at the Maine Science Festival and knew immediately that I wanted to talk to her about her work. Honestly, I could have spent the entire day peppering Jacqui with questions about what she does. It's deeply interesting, important, and for me, has a really cool wow factor. Lucky for you, we kept the conversation shorter than that, but it still has all of those things. I hope you enjoyed as much as I did. Our conversation was recorded in June 2024. So Jacqui, welcome to the Maine Science Podcast. I am delighted to have you here. One of the best parts of this conversation for me is going to be finding out exactly what you do, because unlike many of these conversations I have not actually gotten to see you present about science. I just got to see you host an event, which was enough for me to say, I got to talk to this person, because that was just so cool. So you hosted Science on Tap for the Maine Science Festival this year. So before we get into your current research, I would love to hear about how you got into science and then how you made your way to the States.
Fantastic. Well, Kate, thanks so much for having me on today. I think this is going to be fun. So how did I end up in science? Gosh, I guess I got the bug in, I would call it high school in the UK. I think here we would call it secondary school. And I'm the youngest of three kids. So my sister went to high school and she's a linguist, right? So just really natural at languages. So of course, when I arrive at school and I end up in the modern foreign language department, they've got high expectations because Sally's wee sister is here. And it's absolutely rubbish at languages, just didn't get them. Hopeless, massive disappointment, I think, to the whole department. And the same sort of thing happened with my brother, who was very into maths. And I was okay at maths. I mean, I could do it, but it probably wasn't really my love. But then I found myself in the science department. Physics I could do, but chemistry and biology, I was like, I think I found my home. So suddenly these things that you see day in, day out, a seed germinating or photosynthesis or reproduction, all these things. And suddenly you kind of understood them. And it was just really fascinating. That applied biological sciences really ended up being very, very interesting. And I guess I'd kind of found my own niche where, there were no massive expectations about the clan that had come before me, my brother and sister. And so I ended up thinking maybe science was a good place to be and biological sciences. And it kind of went from there.
So you said both biology and chemistry, which I love. I have a degree in chemistry. I love chemistry, but I learned in college lab work was totally not for me, which was an excellent thing to learn. But I kind of the same thing where I felt like I could see it, right? I could see the atoms, which is not how most people think of chemistry, as you, I'm sure, know. So I'm curious, you had both of those things going for you. What made you lean more towards biology in general?
So it's a good question. I mean, when I went to university to do my undergraduate, then I hedged my bets, I think, and I did biochemistry. Those two words seem to rhyme. Those two words seem to go together. So I ended up with biochemistry. Really, the organic side of chemistry was tough and felt like a lot of remembering, whereas biochemistry, for me, the pathways just kind of made sense. And, you could understand how a chemical was going to be processed in a particular pathway, just because it kind of made sense. And it allowed me to stay in that biology. And I think whilst I was in biology, really overall, the thought of ultimately getting to something to do with diseases and helping about disease was important to me. And so I think that was why, the biology and the chemistry came together. And I actually did biochemistry and immunology at university because I was interested in, in that immune response and how our body tackles infections of whatever variety and foreign matter, foreign tissues and foreign cells. And that really just cemented a sense of, I don't quite know the direction I want to go, but this is really cool. This is really, really interesting.
So did you end up in genetics as kind of a natural evolution of disease exploration? And I'm going to make an assumption here on your age, which happens to me all the time, but it was really starting to become something that you could do, right? When I assume you were starting grad school.
Absolutely. I mean, when you sort of look back at, when the structure of DNA was found out, then, it was kind of 20 odd years from that I was then born. And so by the time I got to grad school, it was on fire. And it was such an exciting field to be in and continues to be an exciting field because it's just an exponential growth in our knowledge and understanding and technologies that we're able to apply. So it's a very, very exciting time to be in this field. At the time that I moved on to do my PhD, I looked at the genetics of resistance or susceptibility to infectious diseases. So that kind of used my biology, but allowed me to start to dip my toe into genetics. And I guess the common theme throughout my career to date has really been genetics, not the disease that I'm studying. So it's using the mouse as a genetic model to study human diseases of whatever flavour. I happen to start with infectious diseases, but in the nicest possible way. I've been around a bit and done a few different diseases in my career.
I'm just going to dive right in. In grad school and currently, or both, are there genetic predispositions to infectious disease in that way? To the point where you can really start thinking about how you address it with treatment and other things?
Yeah, some infectious diseases, yes. I mean, the one that we were studying was a disease called leishmaniasis that nobody had really heard of until the first Gulf War, because it wasn't really a disease that was in North America. It wasn't really in, the north of Europe where I was. But when our service people came back from the first Gulf War, then some of them had been infected with this rather nasty parasite. And so it sort of gained a little bit of traction there. The reason we were really studying it in the mouse, it was fascinating because it really boiled down to this one gene that if you had the inverted commas healthy copy, healthy version, the mice were resistant to this parasite. And if you had the bad copy, then the mouse got the infection. And it was fascinating. It all made sense once we understood what the gene was. But at the time, it was fascinating because the same genetic locus that had been mapped as having this pivotal effect in leishmaniasis also affected a couple of other diseases. So Salmonella typhimurium, which is the mouse version of food poisoning, and Mycobacterium tuberculosis, which is the mouse version of TB. So it was weird because we had a parasite, we had a mycobacterium, a bacteria, all sort of being driven by this one gene in this really strong effect that if you had this very small change in this gene, it would flip the mouse from being susceptible to infection. And so there was a huge push to try and understand what this gene was. And that was sort of the first, I guess, year and a bit of my PhD. So another lab beat us. It was a little bit David and Goliath, actually. And we were most definitely David. A lab that was just better fitted to do that molecular analysis identified this gene. And from a very selfish perspective, I guess for my PhD, it was good because it allowed me to pivot to a different thing. And I had a gene to focus on. I wasn't looking for the gene anymore, and I had a gene to focus on. And so, we found the mouse gene, and then we looked to see what was happening in humans. And in humans, basically the gene ended up being involved in the very early, almost non-programmed response for immunity. So the innate response, which is just the immediate burst response to an infection, not something that's more refined and adapted specifically to whatever the bug is that your body's being faced with. That's why it suddenly made sense that it worked against all these different organisms. The common thread was this protein that is involved in the macrophages in our cells, which are the cells that go around chomping up, eating up all the bad guys. And it really messed up your macrophages if you had a bad version of this gene. So in humans, it didn't turn out quite the same way. So it wasn't such a strong effect in humans. And that was a real abject lesson for me to think, okay, the mouse is a model for human disease, and you need to understand when the results can really be directly interpreted versus when, as a model, you need to understand the limitations of the mouse. Mouse is a mouse and the human is the human. The mouse isn't a small human. And so it was really, really interesting to have that lesson early on. And that's a lesson I really carry with me to this day with all the different diseases and the different disorders that we study, remembering to always be thinking about what you can extract from the mouse and where the limitations of the mouse are.
So is one of the best ways to think about it for someone, honestly, once I start listening to someone talk about genetics research, it feels completely overwhelming to me, partly because I don't have that background and partly because it's still so new. It boggles my mind that we can get so much information from one thing, right? So just bear with me. That was my little promo. So it seems to me one of the most important things to keep in mind then is that a mouse model is not duplicative for a human, but it can start showing you overall ideas, and then you hit either a wall or you learn all you can, and then try to figure out how to either extend that knowledge to prove that it's happening in humans or figure out how it's different so that you decide whether or not it's worth continued pursuit because otherwise it's a dead end. Is that a fair assessment?
I think that's a really lovely summary, Kate. Yeah, I would agree. And I think, when we think about the platforms open in science, then modelling in a mouse is one of many, many options. And the really best studies become this holistic approach where, you start somewhere to get that thread that you kind of want to pull on that idea and then ask, like, what's the best next way of going? Can I do something in cells? Can I do it just with the computer and silicone? Ask all these modelling questions. Do I need to actually see how this change affects a whole body? And if you do need it, what type of whole body? Is the mouse the right organism? Or is it one of the other organisms, yeast and flies and worms and all these other things, and higher order as well. So it's really about always thinking about the question and pivoting to where the night believed to be based on all that you've read and all you've thought about what the next right step kind of is. And I think the mouse fits in a really, really central area there, but it's part of a huge and much larger landscape.
So this is a little bit of a nitty-gritty question, but the example you gave about you found this one gene and it had such an impact on mice, but it was different and not as brutal for humans. How do you study the human part of that? If you could step me through that nitty-gritty, I think it would be super helpful.
Yeah. So for us, with that study, then it actually all started with the humans. So we started from human, not from mouse. And with other members of the team, the humans, they always used to tease us because we were the mice and they were the humans in the lab. So the human geneticists in the lab would go to these affected areas and, with the permission of the individuals, collect blood samples and extract, human DNA from that. And what was really interesting for our disease, for leishmaniasis, was there were some populations where really the disease was not affecting the community. And there were other populations where it was just everywhere. And both of these populations were in areas that the disease itself, the parasite itself, people were being infected, right? They were being bitten by the same mosquitoes. They were getting the parasite, but some folks were developing the disease and some folks weren't. And so it's like, okay, they're all exposed, but there's something going on to make that population, on the whole, not develop the disease, and this population be really severely debilitated by this disease. So that's the thing that says, there's something genetic going on here. We've controlled the environment as best we can. There's something genetic going on here. And so we then kind of pivoted to the mouse to try and start understanding what was going on. And so there are studies that are way outside of my expertise, but, genome-wide association studies where you look at the whole of the human genome and you say, okay, can I see any differences between these populations, this kind of resistant population and this susceptible population? And in humans, there was a huge signal, as it were, for something called your HLA, which is basically the bit on your immune cells that say, hey, I'm me. And depending on what version of that you had, then humans reacted very differently. When we found this one gene that had such a pivotal effect on the mouse, then obviously we looked back in humans and we did find populations with small variants in this gene. So it's not that it was not important in humans. It just didn't have quite such the mega superstar role, as it had in the mice. But in this instance, Kate, we started with the humans and then we went from there as kind of the way we tried to unpick what was going on.
Which is great, right? Because I kind of love this image of you guys going back and forth in the lab. Like, okay, the human folks have figured it all out as much as they can. So they're going to pass to the mice folks. And then you guys were like, okay, go back. Yeah, that's awesome. It's just this whole teamwork thing that I think is really easily forgotten, that's how you advance it. That was great. Super fascinating. Loved it. Let's go to what you're actually doing now. First of all, you have where you're working at the Jackson Laboratory is such a cool center. I think that, the Center for Biometric Analysis just sounds awesome to have on a business card. So that's how you got to the States, I am assuming, besides a little, diversion to Harvard for a postdoc. But what does it mean to do biometric analysis at the Jackson Laboratory? And with that in mind, how much do you actually get to do in the lab yourself? And how much do you have to spend your time being a director?
Yeah. So second question first. The last time I touched a mouse was actually quite a long time ago. So I have a wonderful team that actually does all the hard work. And I really just get the luxury of overseeing that team. So I certainly still think very hard about experimental designs, but I don't do the day-to-day kind of anymore. But yeah, this was my second trip to America. Like you said, I had come after I got my PhD and was in Boston for a while and loved it. And honestly left Boston and thought, been there, done that, got the badge. and went back to the UK and really didn't have any plans to come back to America. And my now boss, Harry Rosenthal, who's a scientific director here at JAX, along with many now colleagues, had this idea that the Jackson Laboratory was really absolutely fabulous at the generation of genetic models of human diseases. But what we were lacking in a coordinated way was a facility that would really allow the systematic characterization of those lines of mice. And that's where the idea for the Centre for Biometric Analysis came from. So there were lots of faculty labs here, all doing their own thing, but there was no kind of centralised place where people could come and say, hey, can we do a kind of a holistic characterization of these lines of mice? So Nadia came to me and said, hey, we've had this idea, and wonderfully, the state of Maine, through a state bond, funded. The good folks of Maine actually voted to fund this. And so the funds were matched by the Jackson Laboratory to build this really amazing facility, which sort of is to try and describe to a layperson, is a bit like the Mayo Clinic for mice. So it's where, you can send your mice in there and they can have whatever characterizations we really, need to do to them. And so, when I arrived, very cold January in 2017, I remember January the 9th, 2017, it really has stuck in my head. There was something called the topping out ceremony, which was pretty new to me, but it's when they take the last metal beam and there was a big Christmas tree on top of it. I'm like, these guys are mad. And so it was hoisted up and there's a fella hanging from it, and popping in this last beam. So this topping out ceremony. So, we built it over the course of the next year. And by the end of 2018, we kind of moved in. And the idea is under this one roof, then we're able to take in these amazing lines of mice that our faculty here make, that researchers all around the world generate but send to mice to look after and maintain in perpetuity. So these lines of mice come to us, and really boiling it down, there's kind of two things we do. Number one, we either really try and characterize these lines of mice to say, are they a good model for a particular disease? Do they show the sorts of clinical characteristics that are seen in humans with this disease? And the second thing is, okay, we've established a good model. Now there's a treatment. Okay, so when the animals are exposed to that treatment, do any of the clinical symptoms that we've observed in the untreated mice start to change so that we can identify whether this might be a good treatment for this disease? And the sorts of characterizations that we do, we have a lot of neurobehavior here. So we're looking at the behavior of the animals, and those sorts of things allow us to study degenerative diseases like Alzheimer's disease or other related dementias. It allows us to study addiction, which is a very strong genetic component to it. It allows us to study aging. It's not really a disease as such, but it's an inevitability, but there are cognitive components to that aging process. So we've got a big focus on behavior. We're able to look at metabolism, how the animal manages its energy levels. That allows us to look at things like obesity, type 1, type 2 diabetes, those kinds of abnormalities. We're able to look at the physiology of the mouse. So really any electrical current going through the mouse, we can try and understand it, whether it's the electricity that makes the animal's heart beat or allows the animal to hear or see. We're able even to look at the electrical currents going on in the brain as the animal rests or performs activities. So we're able to look at all of these electrical activities in conscious, active mice. And topping it all out, we're able to do really high-end imaging of the animals. So we're tremendously lucky that we have a magnetic resonance imager for our animals. So people go and get MRI images all the time, right? And it's this massive, great big magnet with a big hole in the middle of it, and you lie in a gurney and you go and you get your little earphones on, and then, images, whatever, your brain or your shoulder, whatever bit needs to be images. Okay, so we've got a massive, great big magnet with a little 20 centimeter core in the middle of it, which is perfect mouse sized. And so we anesthetize our mice because we won't lie still. Well, a mouse is not going to do that. So you anesthetize the mouse and make sure that it's warm and protected, and then pop it into the magnet, and we're able to image the animal's brain. And then you bring it back out, wake it back up again, animal's absolutely fine. So you can do these longitudinal studies, these studies where you're looking with time at how a disease is progressing or how a treatment is impacting that disease progression. And because we're able to do all of that under one roof, then we're really able to help our researchers here at JAX, because often They have a particular focus. Their specialism is in Alzheimer's and neurobehavior, is on auditory hearing. And they're not necessarily able to pivot and say, there might be a metabolism component, or I really want to do these imaging steps. And they don't have the technology in-house for them. So they send their mice to us, and we do that phenotyping for them. So we're really here as a service to help all of the faculty at JACS with their research and to help external users of JACS mice with their research. So that's, I think, the huge uptick on the upside of my job is I don't study one disease anymore. In any week, we can have 20 disease models coming through very easily, and we're helping all of these different teams take the next step forward in their research.
It is a giant, vast expansion of how you look at mice from when you started. As you were talking, I was thinking, how do you even keep track from hour to hour what you're looking at? I mean, I know you said you have a team, but that sounds, first of all, awesome, right? Because it seems to me that there must have been a wealth of information at JACS from individual faculty members that I don't want to say it was wasted, but it just wasn't being tapped into because everyone's got their own focus, as you said. So all of a sudden you're expanding everyone's knowledge. But how do you keep track of, or do you have to keep track of where the overlap is? Do you know what I mean? Like if someone's talking to you, all of a sudden, do you ever think, we just saw something 3 weeks ago that might really help you out? Is that part of it?
So we have to be really careful because these are our customers in effect. So we need to be really careful there and really respectful of what's going on. Within the faculty members at JACS, it's such a tight bunch of really incredible scientists. I mean, really talented, a bunch of scientists here. And they all talk all the time. So, oftentimes they're coming to us sort of saying, we already learned this from this bit, and now we want to go on to this next bit. So those silos don't really exist to a worrying extent within JACS. Of course, the technologies are really lovely. If we've developed a technology, we're then saying, we go out to the faculty member and say, hey, is it okay if we can use your data to showcase this new technology that we've onboarded? Because we think so-and-so and such will be really interested in using that technology. And we'll then promote that new technology using that scientist and that professor's data. For the externally facing clients, it's a client thing. So their data is their data and not really for sharing. Although because, JACS is a not-for-profit organisation, then the control data, the untreated data, is something that we can reference and we can use and is part of the contract that's made with these external users. And the reason for that is really to absolutely minimise the wasted effort and not waste mice. If we've already run that experiment on a mouse, then we don't want to have to run it again just to know what happened. Obviously, you may have to run controls again. If you're testing a different therapy, then you're still going to have to run your controls so that they're run at the same time. But just for the sake of understanding and for sharing technology, then we surely would never want to waste or use an animal unnecessarily. That's not ethical. So those control data are certainly available.
How about, I guess I was thinking too along the lines of, because you are getting so much sent to you and so much information, both internally and externally, you end up being a hub of information. So when I was saying you could make connections, it's almost like, I'm not saying you use the data, but all of a sudden you have insight on who's working on what, and maybe they don't know each other. Have you found that that has facilitated looking at things faster or differently because you and your team are able to put those dots together?
I think that's a fair point. I think there have been opportunities where we're able to draw those dotted lines. I think we sort of pivot to the kind of other hat, I guess, where I'm principal investigator on a big government-funded, NIH-funded, called the Knockout Mouse Project. And the whole point of that project is to really try and understand the function of each protein coding gene in the mouse. And so the title of the project is building this encyclopedia of gene function. And we're just one of many centres that contribute to it. But those data, the whole point of generating is to generate this resource where anybody in the world can dip into it and use it. We promote it as much as possible. And what we find is our researchers here may have done a particular study that, really focused on, and they're wanting to try and pivot some of the candidate genes they found. And they'll pivot to the Knockout Mouse Project and say, well, hey, they already knocked that gene out. And they did find that there was something related to cognition, or they did find there was something, I don't know, related to vision. So those kinds of shared resources, I think, are another kind of common theme to my career, where I'm really all about open science, and shared science is tremendously important to me, and very much why I find myself in a service position here and, working on that knockout mouse project. It's all about getting the science out there and just facilitating progress as much as possible so that we can try and figure out some of these ghastly diseases, that people and their families are battling with kind of day in, day out. So that's important to me. And although we're right at the very, very, very earliest part of a long pipeline that takes you from gene function all the way through to, this might be an interesting drug target for this disease, all the way through to generating a therapeutic, all the way through to, treatment or diagnosis. We're miles away from that, but we are the beginning of it. And so I still feel involved, even if the end point is many years in the future and with a whole lot of different people having hands on, long after I've released and done my bit, as it were.
I'm really intrigued by the knockout program because as you were talking before about the center, I was thinking, Jacqui doesn't get to do research anymore. And does she miss it? And then it sounds like you've been able to carve out a little piece of that, but also, just as importantly, it sounds like you're also totally engaged in the research that other people are doing, and you recognize how important it is to have as many pieces as possible. Was that a hard transition for you to go into that?
Yeah, actually very, very easy. So, it's important when you decide your career to figure out what you really enjoy and what you think you're quite good at, and kind of not fool yourself. And so I realized really early on that I loved that collegiate component of science and was good at it and was really well organized, which is quite important in my job, as I think we alluded to earlier. And so I kind of allowed my career to take me in a place that, rather than being a master of a single process and a single disease, and digging deeper and deeper and deeper and deeper into one project, I kind of became a jack of all and a master of none, and love it there, so that I'm able to pivot between these different areas of biology and make a really useful contribution and identify, new genetic determinants of particular disease types and know when to pass on the baton. I'm absolutely fine with that. So at the moment, we're from the Knockout Mouse project, we're just in the process of writing up a study where we looked at the physiology of the eye. So we're looking at electrical signals that are created when light hits the back of the eye or hits the retina. So what we did was we looked at just over 500 genes. If you think a mouse has about 2,000 genes. So we're looking for 500. There's a big percentage of the genes relatively. Nobody can do this on this scale. Very few people, certainly. And JAX is one of the few places we could do this. And we tested the electrical signal on the back of all of these different single gene knockout mouse lines. So we took one gene at a time, we turned it off, and we leave all the other genes working. And we say, what happens in this mouse? And that helps us understand the function of that one gene. So most of them were normal, as you'd expect in an unbiased screen. But we identified 33 genes that give altered electrical responses when light hits the back of the eye. And we looked at those 33, and 2 of them are known. I thought, well, that's good, because there's some nice benchmarking that, we're not just generating garbage here. There's 2 that are known. They're already published. One's related to, a disease. One had been found in the mouse. And so, that leaves us with 31 genes that are not known, completely new to the field of vision research, for us to put out there and say, hey, these are important in the mouse. And so we hooked up with an amazing group from Mass Eye and Ear, and they have these kind of orphan families. So they have patients with eye diseases, but genetically they don't fit into any of the usual suspects. And we found a link. So, it's very rare that I'm able to say this, but we've actually found that this patient, they've got a change in one of the genes that we found, and the change looks to be causative. We'll be very careful the way we write it up and just say candidate gene, but the good folks down in Mass Eye and Ear are excited by it, and the next paper will be led by them, and they'll go on and pursue this abnormality that we found. So it's just about knowing when to pass on the baton and say, I've my field of expertise, and now find the right people to get excited about it and say, there's really cool stuff. So that's hot off the press. I was just literally writing this before you and I started talking today, and we'll submit that paper sometime in the summer and confident we'll get it published. It's a really nice piece of work.
So do you have any areas that you're working on right now internally in JAX, not you personally, but I know that JAX has really begun to focus on rare diseases. And I wish I could remember who I was talking to, but the person I was talking to, it was the best explanation I'd heard about why it's so important to work on rare diseases. Because the first initial reaction for lay people is, it's rare, so why are you doing it, right? And I'm going to butcher it, but basically it's because, and this is my take on it, so please correct me if I'm wrong, but very often the rare diseases have far more in common with other rare diseases in the sense of how they're impacting the genome in that one area. It's very similar in other genomes. It just happens to manifest as a different rare disease. So the more you can learn about each rare disease, presumably the more you can learn about all of them because you can help figure it out. It's much more impactful, I think, than is obvious.
Yes, I think so. And it's really about lessons from those rare diseases that can actually be translated not just to other rare diseases, but actually to more common diseases. Because often the rare diseases have a very specific process going on. If you can understand that, then you can take that knowledge and pivot and say, how can I apply that to the more kind of complex disorders? And so it's all about lessons learned. And of course, you help these families that are struggling, with the rare diseases. So you get this wonderful double hit. And Kat Lutz might have been the person you spoke to and our amazing leader of that Rare Disease Translational Centre. And Lon Cardon, our chief exec, came from a rare disease background, so is passionate about rare diseases and looks at the Jackson Lab as being in a truly unique position because of all the strains of mice that we have, all the methods we have to create mouse genetic models for these rare diseases. And then the stuff that we do in the Centre for Biometric Analysis, we're able to really characterise those mice, all meshes together to be a very powerful engine to really make a significant difference to rare diseases. So yeah, very happy about that. And we surely do interact with those folks a lot. And you're absolutely right. A lot of the rare diseases have kind of common threads. A lot of them, there's some kind of cognitive impairment going on. So often different flavors of it, but we're able to study whether it's a socialization or a learning in memory or, what component of that thinking process is affected and really take those lessons learned and apply them more broadly.
I really appreciate you explaining that. And it's a perfect lead into what I wanted to ask you about that. Have you found in the time that you've been at Jackson, you're working on this, have you been able to take some of the knowledge that you've gained and start seeing those connections in a way that has surprised you?
Let me think about that. I mean, where we're at the moment is certainly pulling together those threads of information from individual studies to try and build that really powerful picture of a given rare disease. The truly aha moment that says, we can jump from X disease over to this more common disease. I might not be the right person to have on your program to answer that question. So I can't immediately think of an example of that. I'm sorry.
I'm not sure that was an entirely fair question for a bunch of reasons, including this is still pretty early on in the research.
Yeah.
But I was just thinking because you're a center where you're seeing multiple diseases in ways that other people aren't.
Yeah.
And I don't think there's a wrong answer to this.
No, I think you're probably right. I think the thing that we can do is layer on granularity to those diseases. And a nice example is if we have a disorder where there's some kind of cognitive impairment and maybe a shortening of lifespan, then maybe in humans, we're never going to know whether there was a fertility issue because the patients are never going to reach the stage where we're going to figure out that that's a thing. Whereas in the mice, then we can often build that bigger picture of what's going on in the mouse. And so we've surely had examples there where by studying the mouse model, we're able to think, hold on a second, there's these other things going on in this mouse model, pivot back to the humans and say, okay, might there be something? Might that also be going on in the human disorder? And if there is, that just gives you another thread to pull on to say, I just built that. I've put another piece into the jigsaw puzzle. I built out that better picture of actually the clinical presentations in the human. And those act as clues to what might be going on here. What is the functional defect that's causing all of these things to happen? So we've definitely had examples of that. not necessarily specifically in rare diseases, but in other disorders that the Knockout Mouse Project has sort of generated mutations for.
We have come full circle back to your PhD work of human to mouse and back to human. This is very impressive. Not even planned.
I think it was pure fluke.
This has been a delight to talk to you and to learn just a little bit more about your research. I have a feeling I'm going to be chewing on this over the weekend and come up with all sorts of questions. So I might have to pester you again just for my own edification. But I really appreciate this. I love how well you explained your work in science, that you're not drilling down, but you're doing broader impact. I think it's really easy to forget that that's a key part of science, and we don't talk to those scientists enough. So I appreciate your time in explaining that that is such another great and critical, important role for how we advance science. So I appreciate that, and I appreciate all the work you're doing.
Oh, Kate, it's been such a pleasure. A hoot to go down memory lane of my career all the way back to my PhD, which seems about 300 years ago. And thank you so much for the opportunity to talk with you. I really appreciate it.
Thanks for listening to the Maine Science Podcast. Make sure you never miss an episode by subscribing on your podcast platform of choice. And please leave a rating and review. It will help more people find us and help spread the word about some of the remarkable people doing science in Maine. The Maine Science Podcast is recorded at Discovery Studios at the Maine Discovery Museum in Bangor, Maine. The Maine Science Podcast is executive produced and hosted by me, Kate Dickerson, and edited and produced by Scott Loiselle. The Discover Maine theme was composed and performed by Nick Parker.